back to Cancer Wars. Today we're on episode 5, which is the vascular rebellion strikes. So it talks about the sustained angiogenesis of cancer cells, which is an acquired capability of tumors. So, as an introduction, imagine a bustling city where every building and resident needs a constant supply of food and water to function, which is true. We need to have connections for the supply of resources to the residents of a particular location. And cells are the same. They need oxygen and nutrients for their survival and activity. And each of them must be within 100 micrometers of a blood capillary to receive these supplies because of diffusion. And just think of how wild that is. That means every single cell in your body that needs these is 100 micrometers away, and that's just amazing. So during organ development or angiogenesis, this proximity is meticulously maintained by the synchronized growth of blood vessels and surrounding tissues, and the scientific name for these surrounding tissues is parenchyma. Angiogenesis, the process of forming new blood vessels from existing ones, is a temporary and well-regulated process in healthy tissues. However, in the early stages of cancer development, the rapidly dividing cells and abnormal growths or lesions initially lack the ability to stimulate new blood vessel formation, which restricts their growth. And to overcome this barrier, these neoplasias must acquire the ability to induce angiogenesis. So, how exactly does angiogenesis works? Because it's so fascinating that we're creating these blood vessels and it's having an impact, and it's these small signaling molecules. It's having a relatively large impact in comparison to their own size. So step 1 is endothelial cell activation. And endothelial cells are the cells that line the interior surface of the blood vessels. So we have the the first part is the pro-angiogenic factors and receptor binding. Over here, the pro-angiogenic factors, molecules such as VEGF, which we'll talk about a lot in this PPT, it stands for vascular endothelial growth factor, and these are crucial for initiating angiogenesis and are released by the tumor cells. And VEGF binds to the receptors, for example, VEGFR2, on the surface of endothelial cells. After that, there's a receptor activation where VEGFR2 receptors, they dimerize, which leads to the autophosphorylation of tyrosine residues within the receptors. And this phosphorylation is so important in cells, right? It pops up all the time in bio, and it just creates a downstream signaling pathway. Under many different names for this pathway. For example, the P13K ACT pathway or the MAPK-ERK pathways. And these pathways end with a transcription factor, with the activation of a transcription factor, such as HIF1A or NIF. And then we have gene expression. These transcription factors enter the nucleus and induce the expression of genes that promote endothelial cell proliferation, migration, and survival. Next step is the extracellular matrix remodeling. So over here, the endothelial cells secrete MMPs, which are matrix metalloproteinases, in response to angiogenic signals. So there's a degradation where MMPs degrade components of the basement membrane and extracellular matrix, such as collagen and proteoglycans, and that's what's being shown in the GIF. And then there's a creation of the migration pathway, where the degradation of the ECM by the MMPs clears pathways through which endothelial cells can migrate, and MMP activity also releases ECM-bound pro-angiogenic factors, which further enhance the angiogenic response. The next step is endothelial cell migration and proliferation. Over here we have chemotaxis, where chemo stands, the way I like to remember it is chemo means chemical, and it's moving, and like a taxi moves, but in reality it's chemotaxis is just Greek for chemical arrangement. So over here there's a gradient formation where pro-angiogenic factors create a gradient that endothelial cells follow, and then the endothelial cells migrate towards higher concentration of these factors, moving towards the angiogenic stimuli. And then the growth factors like VEGF promote endothelial cell proliferation through cell cycle signals, and the proliferating endothelial cells expand clonally, contributing to the growth of new capillary sprouts. Imagine them as like branches of the main blood vessel. And then we have integrins, and then the endothelial cell use integrins to interact with ECM and provide traction and guidance for the migration. And endothelial cells differentiate into tip cells and stalk cells. Tip cells which lead the migration, and stalk cells which proliferate and form the sprout. The penultimate step is formation of capillary tubes, which has, the first part is alignment and lumen formation, where the migrating endothelial cells align with each other to form a continuous lining, which is needed. So, the blood doesn't leak. And this aligned endothelial cell forms a central lumen, a hollow space, through vacuole fusion and reorganization of the cell-to-cell junctions. Endothelial cells then deposit a new basement membrane and other matrix components to stabilize the forming capillary tubes. And then newly formed tubes need to become patent, which means open, and regulate permeability to allow blood flow. The final step is maturation and stabilization, which is where we have the recruitment of support cells such as pericytes and smooth muscle cells. And pericytes are recruited by something called PDGF-PB signaling, and both of these, the pericytes and the smooth muscle cells, allow structural support and stability. And then the VEGF and angiopoietins. VEGF continues to support endothelial cell, while the angiopoietins and their receptor Tie2 help mature and stabilize the vessels. After that, junctional proteins like V-caterin and occludins from tight junction spaces between endothelial cells, enhancing vessel stability and barrier function. Next, we're going to talk about the regulation of angiogenesis. So, the regulation is akin to a balanced seesaw with positive and negative signalings to determine the outcome. And it's driven by various soluble factors in the receptors in the endothelial cells, as well as integrins and adhesion molecules that help cells stick to each other and the surroundings. So the first part is the inducers, which are key angiogenic inducers include VEGF and FGF1 and FGF2. And FGF are basically fibroblast growth factors. These factors bind to specific receptors on the endothelial cells called tyrosine kinase receptors. And as you know, kinases are responsible for phosphorylation, and they stimulate the cells to grow and form new blood vessels. And conversely, angiogenic inhibitors such as thrombospondin 1 bind to receptors like CD36 in endothelial cells to block new vessel formation. Integrins, which are proteins that facilitate cell adhesion, also play a significant role, with different types expressed on stable versus growing capillaries. So the critical role of angiogenesis in tumor growth was developed by Dr. Judah Folkman in the 1970s, and his colleagues demonstrated that tumors require angiogenesis to grow using various in vivo bioassays. And in vivo bioassays are experiments conducted on living organisms. In this case, they conducted in mice and found that antibodies against VEGF and dominant interfering VEGF receptor called FLK1 could inhibit tumor blood vessel formation and growth in mice, which leads to development of VEGF/VEGFR inhibitors now tested in clinical trials. Furthermore, numerous angiogenic substances have shown the ability to impair tumor growth in animal models. So the ability to trigger or sustain angiogenesis is acquired through an angiogenic switch during tumor progression. In transgenic mouse models where genes are specifically manipulated, angiogenesis was observed to be activated in mid-stage lesions preceding the appearance of full-blown tumors. Similarly, in humans, angiogenesis can be seen in pre-malignant lesions of the cervix, breast, and skin. These findings, along with the effect of angiogenesis inhibitors, highlight that new blood vessel formation is crucial for the rapid expansion of cancer. And tumors can activate their angiogenic switch by tipping the balance between inducers and inhibitors in their favor. Many tumors exhibit increased levels of VEGF and FGFs while reducing the expression of inhibitors like thrombospondin 1 or beta interferon. For instance, thrombospondin 1 is positively regulated by the p53 tumor suppressor protein, and loss of p53 is common in many cancers, which leads to decreased level of this protein and promotes angiogenesis. The VEGF gene expression is influenced by oncogenes like RAS and tumor suppressors like VHL, which stands for von Hippel-Lindau, which is a common gene that is lost in renal cell carcinomas.
So in terms of proteases in angiogenesis, we study proteases mostly in our digestive system for breaking down proteins. And they do the same over here. So the enzymes that break down proteins and they play a really crucial role in regulating angiogenesis. They control the availability of angiogenic factors by releasing basic fibroblast growth factors, which is BFGF, stored in the extracellular matrix, and they convert plasminogen into angiostatin, a patent angiogenesis inhibitor. And these controls reflect the complex nature of normal tissue angiogenesis and vascular integrity. So, in conclusion, sustained angiogenesis is a vital capability that enables tumors to secure the blood supply essential for their growth. By understanding and targeting the mechanisms behind angiogenesis, you can develop patent cancer